function of the drug analysis unit is to determine if the substances found in an individual's possession fall within one of these schedules. The first step is a screening test. Many times these preliminary tests can be done in the field. Just looking at the evidence may give clues to its identity. Marijuana, for example, has characteristics that can be seen and smelled. A second type of screening analysis is to mix a sample of the substance with a chemical solution and observe a color change. In this example, the blue color suggests the presence of cocaine. The next step is called the selective test. This test may confirm the results of the preliminary test. Examining for crystals under a microscope is one example of a selective test. A specimen of the evidence is mixed with heavy metal salts and viewed under the microscope.
Investigators have science on their side and old evidence with new potential. Now, technicians need only a few bits of genetic material because scientists have found a way to harness DNA's extraordinary ability to replicate itself.
Every cell that you have has the DNA in it. And in order to reproduce the cells, your body makes copies of that DNA.
By getting DNA to copy itself, technicians can manipulate a shred of evidence to power many tests. They do this using a process called PCR, short for polymerase chain reaction.
The main advantages to PCR are that you can obtain a DNA profile from a sample that's 10 to thousands times smaller.
It came from a crime scene. It's older than some of the lab workers. So officers wait, anxious for an answer. And Stedman provides one more than 20 years after the evidence came into police hands.
We were able to detect quality profiles from the sample that was collected from Josephine Otero's sock, as well as the fingernail scrapings from Vicki Wegerle.
When investigators start searching the car for prints, they're looking for two possibilities. Visible prints, known as patent prints, show up easily if a finger has touched something like blood, dirt, or grease. But hidden prints, known as latent prints, need to be developed to be seen. And there are simple tools that can make latent fingerprints appear as if by magic. Powders. Black powder on light surfaces and white powder on dark surfaces. Each scenario creates a high contrast, making the print visible. But how do you create a high contrast when a surface is multicolored, like some magazine covers, or uneven and textured, like a grain of wood? For that, experts can turn to fluorescent powders. Fluorescent powders are applied to surfaces like black or white powders. But in order to see a print with fluorescent powders, you need an ultraviolet light source, which sends waves of color that reflect off the fluorescent powder, making only the print glow. It has taken hundreds of years of trial and error to understand the unique physical properties of blood and the way that blood stains can explain the mysteries of violent crimes. To determine whether or not a dried substance is blood, bloodstain pattern analysts use chemical test strips called hemosticks. They are simple swabs coated with a chemical called tetramethylbenzidine that reacts to the presence of hemoglobin found in blood. Luminol is a powder made up of carbon, hydrogen, oxygen, and nitrogen. When mixed with liquid, it can be applied to surfaces where blood may be found. If there is blood present, the iron in the red blood cells combines with the elements in the luminol, shedding electrons in the form of a glowing light.
You can take 1 part of blood in 400,000 or 500,000 parts of water, and luminol will still find it.